So hello again, everyone. Welcome back. Second session of the afternoon. So I'm going to take you up to 12:15 and give you an introductory lecture on what I'm calling mesoscale and mesoscopic whole brain modeling. And the relevance of those two parts of the title will become clear as we go through the presentation. I want to encourage folks to keep answering questions in the fashion that we had an excellent show with in the first session. And I will do as Itai did and pop back and check on the status of the questions kind of on a regular basis. So just post your questions and we can work together on that. So without further ado, I will bring up my slides. Meta screen share right now. Yep. Yeah. Okay. Well, that's gonna change as I pop over to the slideshow. Okay. Right. So by way of introductions, once again, my name's John Griffiths. I'm the group lead at KCNI in the whole brain modeling group. Also an assistant professor at U of T, and one of the co-organizers of this wonderful workshop, which has been an absolute pleasure so far. I have to say, it was fun organizing, but it's been more fun actually than I expected. Just attending and seeing how the other guys present their work in this context, which is slightly different to the way that we discuss things internally, and also talking with the attendees and seeing how this kind of fits with their professional educational development trajectory. I think it's really been a lot of fun so far, and I hope it continues as such. So here's the plan for this session. What I'm going to do is just talk briefly about spatial scales in neuroscience and the flavor of modeling that I'm going to be talking to you about, which is got these two components. It's on the one hand, or one part is what we call neural population models, and the second part is trying to using neural population models as components in large-scale brain networks, which we kind of use the sufficient term whole brain for this internally, and to some extent the field uses this in general. It's not necessarily whole brain. Whole brain would be brainstem and every part of the basal ganglia and so on. We're kind of, most of brain, strictly speaking, but I think people can allow us that omission, and we're trying to extend as well. So whole brain will be more whole brain as future goes on. Great. So just in a nutshell, before I actually go into that, I want to give you a kind of an idea of what I'm talking about when I say whole brain modeling. So you can see my mouse here. So we start with, and this is now following on from what you heard in the previous presentation, and this is a key way to think about this type of work. It's very continuous with the modeling, the theories, the level of description, the biological understanding that Itai was explaining to you earlier on, and really taking that as the starting point and then going to the next step or a next step. And the next step is principally in terms of scale, right? So the input, in a sense, is taking the knowledge and the understanding that we have in terms of the physiology and the structural organization of neural activity at a local circuit level, and then looking for and trying to define and formulate a reduced description of the detailed physiological, morphological models that Itai was talking to about now. And that reduced description is what I'm calling here, and what one of the things that people tend to call in the field a neural population model. It's not the only term that you'll hear for that, but it's a common one. Other common ones are things like neural mass and neural field and mean field, and I'll be expanding on all of that during the course of the presentation. So so the starting point is to arrive at, formulate, derive, come up with sets of equations that constitute our neural population model, our simplified model of the kind of principal dynamic components of, let's say, a cortical column. Typically, it's a cortical column, but more generally, it's a patch of neural tissue. And then coming up to the middle part of the figure here, then these neural population models give us our basic, our units, our kind of atomic components, in a sense, of a whole brain connectome-based network or neural mass model. Now, I'll be talking about and elaborating on these terms, whole brain and connectome-based, over the course of the talk. But the basic idea is we're talking about a network level description or representation of the brain of the brain at the whole brain scale, or roughly the whole brain scale, with the caveats that I described just now. So the neural population models are giving us the individual units in our network, and specifically, it's a graph, or it's a dynamic network where the edges of the graph, the edges of the network, are provided by what we call the anatomical connectome, or the large scale white matter connectivity, and the dynamics, the activity of the nodes, given by the neural population model. So then the final part is we want to, this is obviously critical. We want to describe the recordings that we make with these models. That's the whole point. And the type of description that we're aiming for, and the type of data that we're aiming to describe, are the measurements that you make at this larger spatial scale, so at the meso to macro spatial scale. And one of the principal types of measurement technique that we use in neuroscience to see and understand neural systems at this meso macro spatial scale is non-invasive brain imaging tools like EEG and MEG. The framework is still relevant to other recording techniques, and ECOG and LFPs, and even calcium imaging. So I will kind of have a focus towards talking about things in terms of neuroimaging, and typically human neuroimaging, but it's worth knowing that this is, that the domain of application for this type of framework does not stop at human brain imaging. So this is the big picture, and really, if you went away from the talk right now, then you would have got the gist. And really, the next hour or so, I'm going to be drilling down on each of these components. I'm going to just flick back over and confirm that everything's okay. I'm seeing Morgan saying there's a problem in the chat there. Is that an issue? Can I get a thumbs up from people saying things are going okay before I continue? All good. Okay, great. Thank you, Itai. We'll continue. Right. So firstly, I have this sketch that I use quite often, and I think it's relevant here. It's not a critical part of the message I want to get across, but it's definitely something that's useful to know about computational neuroscience in general. So this is a kind of schematic of the field of computational neuroscience that I find useful for my purposes, at least. So the basic idea is that computational neuroscience, if you didn't know already, is an extremely broad church, in that the biological questions, the types of people, it basically covers everything in neuroscience and then some, right? Because it covers aspects of machine learning and mathematics and theoretical physics and computer science. So it's, in a sense, it's an even broader field than neuroscience itself. But certainly it's a broad church. That said, that you can roughly summarize the activity that happens in the field of computational neuroscience in 2 broad domains, which I've got here. So on the one half, let's say, I don't know if the numbers are half and half. It will be interesting to try and determine that actually. But conceptually, one half of the domain is research and ideas and theories that are aiming to address questions around basically information processing. I've got a couple of terms here that they're roughly synonymous, but they add a little bit. So information processing, cognition, coding, representation, thought, things like that. Complex behavior. So there's a lot of work going on in various corners of computational neuroscience that are trying to address questions in that area. The other side, which I'm summarizing here in terms of dynamics, is more what you'll be hearing about today. Principally, you'll be hearing about things today that are about things along the lines of dynamics, and this is quite a broad definition as well, but it really spans things that, again, Itai was describing and is understood relatively well at the level of the biophysics of cell membranes, and the activity of small networks of neurons, and both the physics in terms of essentially the electrical engineering of ion channel flow and current propagation, up to things like the network characteristics, nonlinear dynamics of systems that we can understand essentially from a bottom up, in a bottom up fashion, more or less starting at the level of a neuron. We go for we certainly look for abstractions, but it's largely a bottom up exercise, or at least we try and kind of ground things in what we know at the cellular level as far as possible. So those are the 2 kind of lines of research that go on in computational neuroscience, and down the middle is what you can reasonably call an explanatory gap. That is, it's actually quite hard to tackle both of these problems at the same time, and in a way that you could say that bridging this explanatory gap in the middle is kind of the holy grail of computational neuroscience, trying to understand information processing and cognition in terms of what we can really see and observe and characterize in terms of the physiology and the organization of brain systems, brain circuits, etc. Some of the stuff that Itai mentioned in some parts of his talk just now would maybe constitute kind of crossing that gap, thinking about some of the information processing characteristics of cortical circuits. Some of the work that Andrea will talk to you about tomorrow is having a good go at bridging that gap in a few ways, in terms of bridging physiology and cognition. I'm not going to be so bold as to span the 2 domains in the work that I'm going to talk to you about here. So for this presentation, we're on the right-hand side of this figure. So the dynamics box on the right-hand side, there's intended to be a kind of trajectory of spatial scale going from left to right. And I've labeled mesoscale around the bottom right-hand side of the figure. So that's kind of the space we're going to be occupying in the theories and the work that I'm going to be telling you about right now. But this is a useful thing to have in mind when you're trying to understand computational neuroscience, and it's often something that I feel that people who are very new to the field, they have one or other idea about what computational neuroscience is about, and it's definitely about many things, but it's often like much broader than people realize when they're just starting out, let's say. Great. So moving on to something more directly on the line of what we're thinking about in terms of this workshop. I see a question from Sammy there in the chat. Sammy, pop it in the questions tab, and I will come to that and do a round on the questions in a little bit. Okay, so this is a figure that I'm trying to summarize some of the some of the kind of key waypoints, if you like, going up spatial scales. They're going from left to right, or the bottom left to the top right of the screen, increasing spatial scales. And then what are the levels of organization and the terminology, and also the actual spatial scales that are used at each of those levels. So just going from the bottom left, what you heard about in the previous talk, the first hour or so, was these first two levels, like neurons and then neural populations, but neural populations in terms of detailed circuit models. So those are well, the left-hand side in terms of neurons, that would be what we call a micro scale. Micro scale, somewhat well defined because we're talking about systems and structures on the order of microns. And I realized actually that this should say microns, not cubic millimeters, so I apologize about that. So then moving up to past neural populations, but around about, say, between neural populations and local patches. So this scale of on the order of millimeters, this is what's typically described as mesoscopic in the neuroscience literature. And then moving along a little bit further, so then we get on the order of centimeters and tens of centimeters. That's what's typically described as macroscopic in the neuroscience literature. And the kind of top end here on the right hand side is the types of things that we can see with non-invasive imaging. The best resolution generally, for example, with fMRI we can get is down to the level of millimeters, although typically where the actual effective spatial resolution is somewhat coarser than that. But we're kind of solidly in the middle to the right hand side of the scales with human neuroimaging. So another thing to kind of point out here in terms of the scales that I just described there, and this is another thing that is just useful to know when you're consuming the literature and trying to understand what people are talking about, is essentially that one person's micro is another person's meso, and one person's meso is another person's macro. And to be honest, one person's micro is another person's macro. The point is these terms are not well defined. You will see attempts to kind of lay down definitions in certain textbooks and certain, let's say, influential review articles, and maybe that's sufficient for a definition of scales. But in practice, people will use the word somewhat flexibly and somewhat fluently, and particularly meso and macro, or if you like, the interface between meso and macro, and the interface between micro and meso. Those are the really fuzzy areas that the terms are very interchangeable. This is just cashing this out again in terms of one of these figures that you typically see in a neuroscience textbook and in the first lecture and the first day and the first minute of a neuroscience undergrad course. Typically, this figure on the right hand side. It's quite a cool figure from this paper from Sinofsky and co. a few years ago, because it's comparing, and it's probably really out of date already now I think about it, but you see in the bottom right there's the comparison of what was available in the late 80s versus what was available in 2014 when this was published in terms of measurement techniques. The x-axis in this box is temporal resolution, and the y-axis is spatial resolution. So if we just kind of drift over to the top right of this box here, we have fMRI has quite a healthily large size box, both in terms of both spatial and temporal scales, but really a bit insufficient in terms of temporal scales, in that really you want to be over to the left, right? That's in general, you want to be over to the left as far as possible with this scales representation. EEG and MEG are doing pretty well in terms of they're quite far over to the left, but they don't extend down so far. So the spatial resolution is less good than fMRI, but the temporal resolution is better. And just to orient that to the figure that I had just now. So again, these techniques that I've got some little images on the left-hand side: EEG, electroencephalography, MEG, magnetoencephalography. This is what a typical MEG scanner looks like. It's like a giant hairdryer with some liquid helium-cooled superconducting magnetic sensors just sitting around the helmet there. An MRI scanner, which people are probably familiar with. These are all measurements, techniques that will give you measurements around this meso to macro spatial scale. Okay, so onto the first part of the picture that I want to lay out for you about mesoscopic whole brain modeling is neural population models. And what I want to do first is to give a bit of an amusing intuition pump, let's say, in terms of what we're trying to do and why. And this is, it's not a perfect analogy, but it's not a bad analogy. So I'm just going to play this little video here. So these are bird flocks. You could use insect swarms as well, but these are examples of interesting, emergent, complex behavior that happens when you have multiple agents, you could say, or multiple individual elements that are in a set. Well, they're grouping together, they're interacting, but they have what you could call a kind of collective activity, right? So you look at these bird flocks, and they move. They all basically behave like like an organism in a way. They move around. And you could imagine, and I mean, you could actually specifically, you could place, let's say, a spatial average in this swarm, and then think about trying to describe the collective behavior of the ensemble as an ensemble. So rather than trying to think about the individual trajectories of every bird in the flock, just think about the flock as a unit. As an individual entity, and try to arrive at a description that characterizes it at that level. Okay, so that's really what we're trying to do with neural population models. But we're rather than we're working with a bird flock or an insect swarm, we're working with a cortical column, for example, or a large population of neurons. So this is what I'm showing on the right-hand side here. I mean, so what we want to do is we want to go from this high-dimensional representation, where the individual cells in this neural population here would be the analog of individual birds in these flocks, and then move it down to something that is a reduced description. And typically, the reduced description that we're after is something like the average activity over time. So the kind of instantaneous average activity of one or several of the subpopulations of cells that are inside that neural population. So there isn't a direct mapping to the cell types to the bird flock, apart from maybe it could be spatial dimensions. Again, it's an imperfect analogy, but you probably get the gist. Like, we're looking for something like, the centroid of these bird flocks, and taking that as our atomic unit, rather than the behavior of the individual individual birds or the individual cells. So that's part of the motivation and the intuition behind this type of modeling. So I'm going to go through an example of I'm going to go in a little bit of detail on one flavor of neural population model. So I've got a list here on the slide that are some of the commonly used neural population models in the literature and in the field. It's a non-exhaustive list, and there are omissions, and I'm happy to kind of hear people vent about their favorite model that's missing from this summary. But just to say that there are sometimes it's called a zoo, actually. Sometimes it's called the neuron model zoo. It's quite a small zoo, but we're probably looking at on the order of 10 to 20 in total, and maybe 5 really commonly used neural population models. And these can fall into 2 rough description categorizations, which I have here. This is not a perfect categorization either, as in I think of it more as a continuum, but this is often how you see things described as biophysical or biophysically based or biophysically motivated neural population models versus more phenomenologically motivated neural population models. And the difference between these two is along the lines of, on the left-hand side, we're having a go at describing, having the individual units or the individual components of the model being something that you could basically like point to in a detailed model and say, yeah, if I average those, then that would be what this variable would correspond to. Whereas on the right-hand side, there's less of an effort and less of a kind of objective of saying this is a description of entities that you could point to as a biologist. It's more, as the description says, phenomenological. So abstract oscillators, for example, or abstract linear diffusion models. It is still a continuum, and it's a continuum in a number of ways. One reason is that you can sometimes arrive at some of these models, like a linear diffusion model, as an approximation of a biophysical model with certain simplifications, and that's actually a very powerful thing to do. So I'm going to just go through the focus here is going to be on the biophysical model type, and there's again a few in the list there, but I'm going to really just focus on the first one, and this is this family of neural population models. Again, it's not every single type, but this is a pretty good, probably 80% of these type of descriptions. They have essentially 2 basic operations, and it's essentially an operator acting at the synapses, and an operator that's accumulating inputs, and an operator acting at the broadly speaking axon hillock that's defining outputs. Now, in both cases, what we're talking about here is we're talking about or everything we're talking about here is at the neural population level. That is the average of this or that quantity, the average value of that quantity over a population of hundreds or thousands of cells. So just looking at these 2 operators here. So on the left-hand side, again, this operator acting at the synapses is often called, and this terminology goes back to Walter Freeman from the 60s, as a pulse to wave conversion. Pulse here is referring to a firing rate, and wave is referring to a continuous valued membrane potential. So the function that we have here on the left-hand side, this is what's often called a synaptic kernel or a synaptic response function. And this is a function that's describing how the neural population responds to its inputs as a function of time. So on the left-hand side is, on the x-axis is time, and this is basically roughly a skewed Gaussian or gamma function that's with a rapid rise and then an exponential decline that falls off after a few milliseconds. And the y-axis is the response. So it's the level of the membrane potential response in millivolts to a unit input at time 0. So we have this temporal response function, or temporal kernel, sometimes called. On the right-hand side, so our wave to pulse operator, this is now not a function of time, which is to say it's something that's considered to be instantaneous for the purpose of this type of modeling approach. We assume that we have essentially an instantaneous conversion of a population average membrane potential to a population average firing rate. so the function here on the right hand side is doing that conversion. It's saying, you know, the x-axis is the population average membrane potential, the y-axis is the firing rate, and we have this sigmoid function here. You'll probably notice the relationship here between the FI curve that Itai was showing, and there is a kind of concrete relationship that you can derive this from the FI properties of a population of neurons, but you can also derive this from something that's not directly coming from the FI curve of individual neurons. It's actually coming from a simpler description of neural responses that's more like a leaky integrating fire model, or actually more specifically, it's just on-off. And I'll talk about this on the next slide, actually. But this sigmoid is what's providing our wave to pulse operator. So defining how the output, defining what is the output signal of the neural population instantaneously in time, given the population average membrane potential. So just kind of drilling on that, down on these a little bit more. So the synaptic kernel on the left-hand side, this is something that's often, well, it has two equivalent mathematical formulations that are both useful to know about if you're thinking about this. I'll just describe it briefly here. It's really not essential for people who aren't, you know, really getting into this modeling to know it to understand it in too much detail. But the idea is that, you know, for the convolution or the integral representation, we have our function h. So that's what this function is here. This h, and the way that this acts on the input is by way of a convolution. And a convolution is kind of like a moving window cumulative multiplication, which is represented with this integral here. So where t is tau is kind of the lag. So it's actually kind of projecting this back in time. So you're kind of moving along in t, and then you're saying for a given time window, go back, and then I'm going to accumulate the or convolve the incoming inputs with this function to give me my population postsynaptic membrane response. So we have the integral representation here, but when we're simulating these this system, it's actually more convenient to use differential equations. So there's a corresponding differential equation representation of this integral here in terms of a second order system, which we can rewrite as a pair of coupled first order differential equations I'm showing here. And essentially the interpretation of these is that I is the membrane voltage, and V is the derivative of the membrane voltage, which is the we could think of as a current. So this is when we're working with these systems numerically, then typically we'll be writing down equations that are more like this, but it's easier to understand what's going on from arguably, and typically people think this, and that's why they describe them this way, easy to understand what's going on in terms of the integral form of the synaptic kernel. On the right hand, so turning to the second thing, the output function, the sigmoid firing rate function, we have, so this is just the expression for the sigmoid here. With R as the slope, and so what I'm showing here on the right hand side is what I started describing earlier on, but it's easier to show with this actual figure here. So this red step function here, it's called a Heaviside function, and this is what this type of model is basically assuming that this is the description, or the assumed description by this model, or by by this type of equation of what's going on at the level of single neurons, which is the probability of them firing, or the probability of a neuron at a given point in time being active, as in being like firing an action potential, is a function of its membrane potential, and it's a step function of its membrane potential. So the point where the red line the steps here is the threshold, firing threshold. So we're saying when a cell reaches its threshold, then it's firing. Okay, that's what this function is showing here. Now, if we have a number of large number of cells that we can kind of describe in this way, but have slight variation in essentially the location on the x-axis of the step of the inflection, then and then we average those. So then we'll get that will average out to the sigmoid here. So the reason for that is the justification for that, the thinking behind that is that we have a number, say we have, doesn't really matter how many, but a sufficiently large number of cells to achieve a smoothed average. So let's just say a few thousand, with slight variations in the point in their presynaptic, their membrane potential at which they fire. Now, the reason for that variation could be due to a number of things. Could be due to different physiological properties of the cells, different wiring properties in terms of their inputs, different just current state in terms of what their incident incoming inputs are. But we just assume that there is variation in for any given cell, the point at which it will fire, given its membrane potential, and that variation is normally distributed around some average. So we'll have a normal distribution or a variety of these red lines that are kind of, a little bit further back to the left, a little bit further over to the right, and we take an average over those, then actually that works out analytically to a sigmoid function. So that's the rationale for using the sigmoid firing rate function at the neural population level. Now, it's interesting that the same function or a similar function, it comes up all over the place. Itai mentioned, the FI curve that he showed was a kind of rectifier. He said it was a rectified linear function, but sometimes those are described in terms of sigmoids, just at the neuron level. And this is also used in artificial neural networks. And in fact, the first formulation, the first use of this formulation in the late 1960s in the context of neural population modeling was done more or less exactly the same time in some of the very earliest developments in the mathematical formulation of artificial neural networks. So the chap who came up with the use of the sigmoid function in neural networks was Grossberg in, I think, 68. And then roughly the same time, both Freeman and Wilson and Cowan came up with a similar type of function on the basis of a more kind of physiologically based argument along the lines of what I just described, of what the population average firing rate function would look like. So sigmoids, they really kind of come from lots of different locations, come up in lots of different contexts in biological systems in general, but specifically in models of neurophysiological systems and neuron-like systems. They clearly have important properties in terms of making stuff work, and it's obvious what it is. It's saturation, primarily, keeps things relatively stable. So wrapping up on the picture for neural populations, and I will finish up on neural populations, and I will address some of the questions we have, and then I'll turn to networks. So the picture that I just described was the key components of a neural population model for a single neural population, the wave to pulse and the pulse to wave operations. So typical neural mass models, they consist of a few elements, a few of those neural populations that have those input and output functions. And the most common flavor of that is some combination of excitatory and inhibitory populations. So this figure on the right hand side is a very nice schematic from a very nice review article from David Liley from a few years ago that's showing some of the many of the neural mass model schematics that are used in the literature that are, as you can see, kind of small motifs that use essentially minor variations in the number of excitatory and inhibitory populations they're describing and the topology of the wiring between the excitatory and inhibitory populations. So the kind of classic one, or one of the classic ones right at the top here, is a Wilson-Cowan. And by the way, that we also have in this figure, we have different, these are also grouped by the function of the postsynaptic response, the type of the postsynaptic response. That's a little bit more detail than we need to get into here, but it does follow on from the response functions that I was showing you on the previous slide. So the classic, really like the number one neural mass model, I think you could reasonably say, is the Wilson-Cowan equations, which is a system of integrated differential equations that describes a cortical column, slab of cortical tissue in terms of an excitatory and inhibitory population that that project to each other and also project back on themselves. Some of the other schematics that have come up are, so a common one, and that I'll talk about in a second just now, is a Janssen-Ritt model, which has two excitatory populations, one inhibitory population, and a limited schematic between the two. So we'll see this in a slide or two, but the middle excitatory population here is the pyramidal cells. And essentially the pyramidal cells have bidirectional exchange, bidirectional connectivity between one excitatory interneuron population and one inhibitory interneuron population. And then other folks have extended this in, like numerically and theoretically non-trivial ways, but you can kind of see what they're doing, right? They say, okay, we're going to have an extra excitatory and extra inhibitory population. We're going to add questions between, add connections between where sometimes they're missing, and those can have quite profound effects on the behavior of the system. And another one that's kind of falls roughly into this class is a Robinson model, which I've used quite a lot, especially recently, which does the same kind of thing, but it has excitatory and inhibitory populations that are in the thalamus rather than in the cortex. But the basic framing is the same. They're excitatory and inhibitory populations. There's a bit more of a delay between the cortex and the thalamus in this model, but otherwise they can be understood within the same general formulation. And one of the key things about these type of systems, and this is, I possibly would even put this to my slide when I said that, like, take-home messages, things I would like you to take home, is that one of the most important things about that, that we kind of learn, and the understanding that we can get from this type of simplified formulation is that we can study things like the emergence of oscillations, right? So in systems of this kind, not restricted to biology, and we're thinking about these things in terms of biological systems, but we're also thinking about them in terms of general control systems or general electrical engineering systems. Oscillations occur under certain conditions. One of the prerequisites of which is some form of negative feedback. So in the physiological context, the negative feedback is coming from the inhibitory population. So it's either inhibiting itself or it's inhibiting another population, like the excitatory population. So yeah, from a physiological point of view, we think about that as inhibition. From a more control systems point of view, we think about that as negative feedback, same thing. But that's one of the key functions in these type of roles that the dynamic between the excitatory and inhibitory populations is resulting in. But that's one of the kind of insights that we can learn. And, I often find that people talk about inhibition a lot as inhibition, right? As in, like, the principal role of inhibition is to inhibit things, is to keep the activity level down. And that's probably the primary function of inhibition in general, inhibitory activity in general in neural populations, but it's not the only function, right? Another function is to generate oscillations, and it's often overlooked when you hear discussions of the potential impact of disease-related changes to inhibitory cellular activity, for example. It's going to affect oscillations, not just general inhibition level. Great, so the Janssen-RIP model is what we will use in the tutorial this afternoon, and I will not go into it too much here. I've given you all of the background just now, but just to say with a picture that this is the system, this is the reduction of the cortical column that we're talking about here, and it's laid out on a roughly cortical column kind of background to hammer that home. So we have our pyramidal cell population, we have our excitatory interneuron population, which is described as spiny stellate cells here. There are obviously, these aren't the only excitatory inhibitory cell types or cell types that you could enumerate. There are some missing, but it's trying to kind of capture a lot without trying to capture everything, if you like. So we have our 3 cell types, 3 neural population types, and these are all connected to the pyramidal cells. So more on that later in the tutorial part. Finally, in the neural population model part of this talk, I mentioned oscillations just now and how inhibition is a key for negative feedback is kind of a key prerequisite for generation of oscillations in general. Now the way we study this, and we will see this in the tutorial later, both in the first tutorial with Itai and the second tutorial with me, is the typical thing to do is to basically simulate neural activity, integrate your differential equations, generate simulated time series, and then study the frequency content of the simulated time series as you would do experimental data. And that's the easy, that's the easy way to do it. And when you're working with systems beyond a certain level of complexity, then that's the only way to do it. But there is another way to study oscillatory activity in neural mass models, which is very powerful, and that's to develop analytic expressions for the power spectrum. So in a nutshell, what you can do is you can take, I haven't shown you very many of the actual equations that are used here, but you can take one of these model types and we can set certain conditions where the system is in what we call a stationary state or a steady state. And then that is, and we set a few other conditions. We need to linearize, and we need to linearize the system, and then we need to be able to come up with an expression for what's called the transfer function. When we have the transfer function, we can get what's called an analytic power spectrum. An analytic power spectrum is basically a direct mapping from the parameters of the model, which would be, for example, the excitatory inhibitory gains, or the weights, if you like, of these two of the synaptic inputs from one population to another population. We get a direct mapping from the values of those parameters to the power spectrum itself. And this is very useful and very powerful. It's useful and powerful for theoretical reasons. It allows you to do certain kind of systems analysis, study the poles, understand the system at essentially a deeper level than you can do with what sometimes called brute force simulations. And it's also helpful because it's quick. It's really quick. It's just a linear operation. So it's extremely powerful for searching large parameter spaces, fitting data. It's actually certain types of data fitting you wouldn't even try to do with a sufficiently complicated neurophysiological model because it just takes too long to run and the parameter space is too complicated. So there's a lot you can do with linear models. We're not going to look at linear models, but it's where I just wanted to give it a flag because it's one of the things that when we're using this reduced, this kind of simplified description, and I want to be the first to point out that this is a dramatic simplification of the way that neural activity functions, right? No one's going to argue with that. Question is it sufficient for learning certain things? One of the things that we can get when we simplify things dramatically is there's a broader realm of understanding that's available to us that are using the mathematical techniques from engineering and also in some cases say nonlinear dynamics that are kind of bread and butter really for people working in electrical engineering, trying to understand the properties of circuits. So there's a broad domain of tools for characterizing systems that we can have at our disposal if we can formulate things down to a simple enough but not too simple description of the system at the level that we want to study it at. Great. So I'm done for the neural population section, and I'm going to go over and see how we're doing on questions. 8 questions. Got another half an hour in here, so I think we're doing okay. Right. So first question is from Itai. Why do you need two excitatory populations in the Janssen-Rit model? It's a good question. I actually don't have a good answer for that off the top of my head. So the Wilson-Cowan equation does not have 2 excitatory populations. It's also slightly different mathematically to the Janssen-Rit model, or the precursor of the Janssen-Rit model from the 70s. But I think that the answer to the question is it's included because it was considered to be an important part of the biology to include in the circuit. Now we could have a discussion, and we probably should have a discussion as to whether that's a critical thing to include or not. But that's the rationale for it being included. Now, in terms of the impact that it has on the behavior of the system, I haven't studied this extensively myself, but the principal thing that is going to allow is kind of an extra gain control, right? So it's kind of an extra level of positive feedback so in terms of the properties that would result from having that extra excitatory population. But it's a good question. I mean, one of the questions that's posed more often is, do you need 2 inhibitory populations? And same questions apply in terms of what its impact is on the system. But from the sketches that I showed you, that's something that's kind of often, and I've seen this myself from reviewer comments and so on. It's like, why do you not have this extra inhibitory population? And it's really important. So on. These are the kind of things that get debated: is how many to include, and what do you get from adding in extra ones. So partial answer there, Itai. We can pick that up later and try and improve on it. Okay. So Sunny says, there's a lot of overlapping concepts, tools, formulae between, are there a lot of overlapping concepts, tools, and formulae between neural population models and other population models like work in ecology, to your flock analogy, and human populations in urban planning? And this is an excellent question. The answer is a resounding yes. And to give 2 concrete examples. So in my opening slide, I had this sketch of the field of computational neuroscience, and I mentioned that on the right-hand side, there's kind of a trajectory in terms of spatial scale, and one of the ways that's manifest, or one of the terms that I had on the figure there was statistical mechanics. So the inspiration for the type of reduction that we're looking for in this type of approach, neural population modeling type of approach, is actually the most direct influence is really coming from statistical mechanics and things like mean field description of the behavior of gas particles. So I was talking about bird flocks. Arguably, gas particles is a better analogy, but what you're talking about there is things like the pressure of gas in a room or the temperature. Okay, so things that are terms and quantities that are have been in the canon of theoretical physics for hundreds of years, or at least going on for 200 years, in terms of statistical mechanics. So descriptions of gas and fluid behavior that is abstracted from the individual atoms. Okay, so that's definitely part of the general inspiration, and a lot of the mathematical techniques in terms of the derivations, they really come directly from the way that mean field descriptions are derived in statistical mechanics. The second, and I know that wasn't necessarily exactly what you were getting at with that question, because you were talking about ecology and so on, but that's definitely like an overlap in kind of the idea of a mean field in general. Now, the second answer is that the Wilson-Cowan equations, which, as I said, is really like the poster child of neural population models, those were inspired directly by the Lotka-Volterra equations, which is a model from ecology that is describing the oscillations that occur with predator-prey cycles. So you have, I think, foxes and hares, or something, and lynx. I can't remember which one Lotka and Volterra were studying, but foxes and hares is a common one, right? So you got the predators and the prey, and like the population, the number of predators in the population. If there's too many predators, then, well, if there's more predators, then the prey go down. But then if there's not enough prey, then the predators die. So then the prey go up again, and so on. So this kind of oscillatory behavior, this has been studied in ecology for a long time, and this was the inspiration for Jack Cowan's formulation of the equations that bear his name. And it's interesting. I mean, that's kind of what excitatory and inhibitory populations are doing. They're pushing and pulling against each other, and they're kind of, it is a bit like two populations of organisms that are kind of, antagonistic to each other. So that's interesting. If you want to read more about that, then if you Google Jack Cowan stuff, then you'll find one or two YouTube interviews which are amazing, and it's just blows your mind, like who those guys were talking to in the 50s and the 60s. It's like Wiener and von Neumann and all those folks were just hanging out. So I'll wrap up on that question. Srijoy says, are the population models supposed to be comprehensive or as detailed as the biophysical model described by Itai? Absolutely not. I think I stated this reasonably clearly in the last few minutes, but no, they're not supposed to be. The question is, what can we get away with, right? Or how much do we need? And as simple as you can, but as simple as it needs to be, but no simpler, is a reasonable heuristic. But I think life is more complicated than that. So it's really a question of what we're trying to achieve in terms of the detail. But this is an active research question, is how much should be included and how much is too much of an omission. But no, they're not intended to be as comprehensive. Although I'll say one more thing on that, Srijoy, is that one thing that I'm looking into, and it's interesting, Itai has these four populations. He has the SST, the VIP, the pyramidal, and the what's the other one? Forget right now. I haven't seen any neural mass models that are explicitly describing those cell types. And that would be an interesting thing to think about putting together. Yeah, PB, thanks, Itai. And we'll probably do that over the next few years. Okay. Another question from Srijoy. Does this flavor of computational neuroscience require a strong background in mathematics? Excellent question, especially for this workshop. And this is a common question that I have a pre-prepared answer for. Is it yes and no, right? So essentially, you can definitely come in and do substantial amounts of high quality work without really understanding any of the equations you're working with, as long as you understand the biology and understand what's going on at a conceptual level and are diligent in confirming that you know enough. So you don't need to be writing out the equations. You certainly don't need to be deriving the equations. But you need to know certain things about what an equation is and what doing a simulation is and stuff like that. So, and strong background in mathematics to say, I don't have a background in mathematics. Itai doesn't have a background in mathematics. I don't think half of the people in computational neuroscience have, strictly speaking, a background in mathematics. They have a really kind of rich tapestry of backgrounds in various fields that add to their kind of USP as a scientist. And if you do have, or if you want to develop your mathematical expertise and the kind of level of that, the extent to which that features in your work, then there's room for that as well. Obviously you need to pick stuff up if you haven't picked it up already, but you can. There's a place for everyone with more or less any level of background in mathematics. Probably the primary common denominator is, unsurprisingly, you need to be kind of somewhat competent with computational work, which I'm pretty sure everyone here would be. So Sammy has some interesting follow-ups there, but we can talk about those in the break. Okay, moving through these a bit more quickly now. Yes, the parameterized sigmoid in the neural mass is analogous to the FI. Thank you, Itai. Yep, that's good. Next one, Akshay. Are the shape of these two curves well accepted, or is it possible that it may change? So the sigmoid activation function was studied by Freeman in the 60s, and that was in the olfactory bulb, and that was the motivation for using that function in the last 40 years of neural population modeling. So I know that much, is that in the early work, there was a kind of somewhat solid basis for saying, okay, this is the correct level of description. This is the correct equation. It matches experimental data. I haven't checked what people have said about that for the last 40 years. And there was an interesting paper that came out a few months ago from some folks in London that were kind of studying the accuracy of Wilson-Cowan equations with high throughput neural recordings, and they had some interesting kind of statements to say about their accuracy at various levels. But yeah, I think broadly yes, but it's an interesting question what the current state of affairs is, because things change over the decades, and I don't know what the 2020 answer to that question would be, but I'll definitely try and find out. Right. This is a question from Zuri. Is the middle graph a result of models around it? If so, what's the difference between those models? I guess this was for the Liley diagram, and that diagram, there was no particular relation. No, the middle graph wasn't a result of the models around it. They were just different categorizations. So we can follow up if that wasn't exactly what you were after there. Itai has a comment. PV neurons have been shown to mediate cortical gamma oscillations. Excellent. Thanks for that observation. 2 more questions. Is there an established electrical circuit that illustrates the behavior of a neural population in terms of its oscillations during depression? No, there is not. There is no established anything in depression in this line of work. It's very much active research areas. Something I'll come to near. I mean, other areas have been studied relatively more, such as epilepsy. There's a lot of work in modeling, trying to understand some of the mechanisms of epilepsy. But depression and everything in psychiatry is really pretty much open territory for a lot of this type of modeling. And Andrea will have more to say about this tomorrow. So there's not a whole lot understood about the circuit properties of depression at this point in time. This is kind of our job. In all of these models, the focus was on cell type, not extracellular environment. Is there a reason to ignore the extracellular effect? By extracellular effect, maybe you mean ephaptic coupling and local fields. If that's the case, then yes, that's ignored. And there are voices coming from various corners that are arguing for including ephaptic effects in particularly micro level models, but it's not something that is really discussed much at the level of neural population models. And really the reason for that is we've really kind of abstracted from space quite a lot. With we're talking about averages, right? So an average of the activity of cells in a population, this is not a physical quantity anymore. It's a statistical quantity. So, and we're not encoding space in any way. So we're really abstracting from space, and in a sense, we're abstracting from physics as well. Zora, you mean extracellular geometry and fluid properties? Yeah, same applies. We're abstracting from space. So if you're talking about the macro level extracellular space or macro level geometry, like Sami was asking about earlier, then yes, we think about that, but that's not really the level of the neural population model. So this is an interesting question, and we can follow up on that later and drill down on that if you like. Excellent. So back to the show. Okay, so what is this thing whole brain modeling? Excuse me 1 second. So I had the opening slide that gave you the elevator, the 30-second version. And what I want to do now for the second half of this talk is just drill down on a few of the details of that and a little bit of the terminology. So this slide is just summarizing what I also showed you at the start, which is that what we're doing is, and it's kind of back to the front really. On the right-hand side, we have our neural population model. Actually, the left one is a schematic of a Wilson-Cowan equation. The right one is a schematic of a Janssen-Ripp model. These are both, somewhat simple neural population models that are describing in a reduced way the neural population or the cortical column that's in the middle, and then each of these units is forming one node in a network that is defined by anatomical connectivity. So where is this anatomical connectivity coming from? So what I want to do here is just say a little bit about how this is studied and understood in terms of brain imaging, principally, but also a little bit about connectomics, which some people may not have heard or know much about as a concept. So the connectivity of the cortex, this is a coronal slice of a T1 weighted MRI image here, and the little square is probably around about the right dimensions of what maybe it's a little bit wide, but that's what we're talking about. We're saying like a piece of brain, a patch of tissue. That's where these neural populations, these cortical columns, are sitting. And the connectivity laterally is typically understood to be what we call translationally invariant, which is to say, so these are these kind of white things here. These are showing, these are actually from Blue Brain sketches, but they're showing these hexagons here as adjacent cortical columns. And the connectivity between the adjacent hexagons is what we think of as roughly translationally invariant, as in any one of these hexagons has the same connectivity profile to its neighbors as any other one of these hexagons. However, the long-range connectivity is not necessarily as simple. So what I'm showing here now, again, we have our coronal slice, and then the middle here is showing it's actually a tractography reconstruction showing basically what's some of the white matter fibers that are going to be coming from the different parts of the cortex that you can see on the extent of the cortical surface ribbon. So this has been understood and studied for quite a while, and with certain types of dissections in postmortem brains. The top one is called a Klinger dissection. Nowadays, we use non-invasive imaging to study this kind of thing. So this picture on the bottom here is from non-invasive diffusion MRI tractography, and the little bundles here, the little colored things, are essentially different white matter tracks that project from between different parts of the cortex. That we can identify and kind of trace these streamlines non-invasively with diffusion weighted MRI. So the general idea, or one of the key ideas that's been driving this line of research forward, is this idea of the connectome. So this term, the connectome, it's an analogy, possibly a loose analogy with the genome. But the idea is the connectome is a complete map the idea of connectomics is to study the map of connections between brain regions in the same way that one of the kind of the kernel of genomics is to study the human genome and the map of base pairs in DNA. So that's the idea behind connectomics, and this idea really picked up in, the phrase and a lot of the narrative around this picked up around the early 2000s, and it was coined by some folks in neuroimaging, Olaf Sporns and Patrick Hagman, kind of independently, and it also picked really has been really influential at the level of micro level neuroanatomy as well. So some folks, Jeff Lichtman on the right hand side and Sebastian Sung, who is now head of research at Samsung, I read. These are the folks who've been studying the same question, trying to get a detailed map of all of the connections of a brain, but at the level of essentially I think a piece of mouse brain that would fit inside the middle part of a dime, so just about a square millimeter. So these two lines are kind of really been going in parallel, the macroconnectomics and the microconnectomics. With the macroconnectomics, we're not studying connectivity at the level of synapses, but we're studying at the level of large-scale projections. So the general approach is to break the brain up or divide the brain up into what we call parcellation. Parcellation is a like modern-day Brodmann regions. So, maybe you've heard of Brodmann regions, BA6, BA22, etc. These days we talk about parcellations. So we basically it's a list of brain regions, or kind of assignment of locations on a brain, these colored patches here on the top left. And definition of these in terms of brain regions. This is quite an old one. This is AAL. And then, so the pipeline is to we have our parcellations, we compute tractography as I just described, and then we look for the streamlines, so the little spindly things in this picture that interconnect each of our parcel pairs. And when we find streamlines, we count them, and those give us entries in our adjacency matrix or our anatomical connectivity matrix. And that's the methodology. It's represented nicely here in this little movie. So we go from MRI image, and we can take our tractography reconstruction. We combine this with a brain parcellation that is showing here, and then we summarize this in terms of a graph. And this is a graph represented in 3D space now, but it's really an abstract object at this point that's saying, okay, there's a certain level of connection weight between each of the entries or each of the brain regions in my parcellation. That's the methodology. There's been a lot of work on this in neuroimaging. Human Connectome project kind of led this forward, but it's really, it's a core part of the neuroimaging community now. It's like more than half of the work at HBM, which was a conference a few weeks ago. It's always now at least half of the work you see is about connectivity and connectomics one way or another. So the paradigm of whole brain modeling is using the network that we're studying is the anatomical connectome, and the phrase that we often describe these type of models is a connectome-based neural mass model. I'll skip this, but there's also connectome-based neural field models, which have more of an explicit encoding of space at the level of individual nodes rather than point processes. And a tool that we'll be using later on that I'm a little bit involved in the development of, and that's pushing forward the software infrastructure for this type of modeling is the virtual brain platform, which is a pretty nice toolkit. Again, we'll look at a little bit later on today. And bottom line, so just an example of the type of thing that we can do with these connectome-based neural mass models is this work that I've been doing for a little while with Jeremy Lefebvre. And what we're doing here is we have our connectome, our neural population model that's sitting at one of the nodes in the graph. And this generates rhythmic activity, which gives us a good representation of the alpha rhythm. So alpha frequency activity at the level of the whole brain. So the figure on the right-hand side is the activity generated by this connectome-based neural mass model when we run the simulation forward, and the activity of each of the nodes is painted on parts of the cortical surface. And you can see, if you just kind of track in time, that we have these they're not spatial uniform, so there's spatial structure there, but the majority of the oscillations are waxing and waning on the order of 100 milliseconds. So this is a connectome-based neural mass model of the alpha rhythm, which is the type of thing we can do with these models. So what I want to do in the final part is go through a quick survey of some clinical applications. I think I will just blast through this, and then I'll come and I'll take the questions, all the questions at the end. Because we're a little short on time. We have another 5 minutes, if I'm right, before our next break. So just flying through some quick examples of connectome-based neural mass models in action. It kind of fits into the genre, but it's not quite connectome. It's more like large-scale network, is modeling of auditory attention mechanisms. And Itai actually mentioned this earlier on, and he also mentioned that this is something that Andrea will be giving us some, doing a deep dive on tomorrow. So I'll echo his sentiment in not going into this in too much detail, but just to say that the type of model that has been used quite extensively to describe the oddball response to, or the auditory response to unexpected stimuli, is a version of a neural population model, not with hundreds of nodes, but with just 5 nodes. But each of these nodes in this auditory attention network is a Janssen-Rit neural mass model, or exactly of the type that I've described earlier on. So Andrea will give you more information on this, but this is a kind of preface that this is a link between what you've been hearing about today and what you'll be hearing about tomorrow. A big domain that's been quite successful on the clinical side, and I mentioned earlier on, is modeling epilepsy. So this is an example from some a model, but it reproduces empirical data quite well of the generation of an epileptic seizure. So each of these little dots here is a depth electrode, and the traces on the bottom are the traces, the LFPs that are recorded by each of the contact points on these depth electrodes. But you can see as this spans forward in time, the spreading of the seizure from the initial seizure zone through to a number of the other brain locations. So this is what seizures look like, high density, high frequency sampling, spiking. And when they spread, that's a problem. So one of the things that's been studied quite intensively by some of my colleagues in France, Victor Yersa and co, is the behavior of the transition to seizure. And they developed a model they call the epileptor, which kind of cycles through periods of high frequency spiking seizure activity and reproduces a lot of the phenomenology of seizure behavior. And what they're doing is they're using this in a connectome-based neural mass modeling context to try to provide recommendations for surgeons in terms of resection strategies. And the basic idea being that they fit the model to data to be able to identify what they call the epileptogenic zone, and what in red here, and other parts of the local graph, the parts of the connectome that constitute what they call the propagation zone. And the seizure initiates in the epileptogenic zone and then propagates through the propagation zone. But the key idea is that in order to prevent a grand mal seizure, so global seizure spreading, ideally you want to hit the epileptogenic zone and resect that tissue, but sometimes that's not feasible for various reasons, in which case you might be interested in resecting the parts of the propagation zone tissue if, in this context, the model suggests that will have an effect in terms of preventing large seizure propagation. And this is really fascinating and exciting and kind of heartwarming application of this type of modeling framework. It's undergoing clinical trials in France at the moment, and it's really going quite well. So I encourage people to take a look at this work if they're interested. Another clinical area that's quite interesting, quite close to my heart, because I used to work in this area, is in stroke. So the gray image here and the black image here. So this is what it looks like when you have a stroke in an MRI image. This is a representation, this big gray blob of what a lesion would look like in relation to the white matter. And in terms of a brain network, then you can think about this as there being nodes and edges being removed, right? So it's pretty kind of easy in a way to understand how you would model that from this point of view. And some folks in the US have been looking at this, trying to fit these models, and have been able to associate some of the parameters that they fit with these models to neuroimaging data, and associate them with recovery in terms of motor assessments on the course of months after the patient's had the stroke. So it's promising in terms of potentially a biomarker for recovery success at the level of motor recovery. And then finally, so an area that I'm working on at the moment intensively, we'll see a little bit of this in the tutorial session this afternoon, is brain stimulation. So in a clinical context, brain stimulation is a therapy that's used for treating depression for patients with treatment-resistant depression, specifically, who don't respond to medication and have typically several bouts of failed medication and need another solution. So the methodology is basically go in with a big magnet, big coil like we have on the top left-hand side here, and stimulate the brain transcranially. It's called transcranial magnetic stimulation. So we can use other tools to estimate the the electrical fields that are induced in neural tissue as a result of this application of magnetic stimulation, also electrical stimulation. And then the job for neural mass modeling is to say what happens then. Okay, and there's two things that you might be interested in, principal things you might be interested in this context. One is, where is this activity propagating to? Right, so when you stimulate in dorsolateral prefrontal cortex, which is a typical stimulation site, are you hitting other parts of the network via indirect polysynaptic pathways? So we can understand that on the first order. We want to understand that in terms of the brain network, the anatomical connectome. But after that, we want to know, using physiological simulations, and this is the context where you really need a whole brain model to study this type of large scale propagation resulting from brain stimulation. What additional contribution over and above the basic neuroanatomy, the basic wiring diagram, does the physiological response of the neural tissue to initially the stimulation, but then subsequently the polysynaptic propagated stimulation? So this is something that we're actually working on in my group quite intensively, is trying to map out and understand the immediate responses and the kind of downstream network regions that are targeted by brain stimulation in one or other precise coil location in the context of, say, specifically a DLPFC stimulation. Or if you're going to move over to a completely different brain region, then what can we understand in terms of the predicted and potential response of downstream brain regions in a different site that maybe has not very much research around it. So this is the type of thing that we think doing modeling can really contribute to, is some part of this discovery science regime where we can try things out in silico, as Ito was describing, that it cannot be done experimentally, or at least cannot be done with the scale and with the ease that we can do in a computational context. And secondly, and finally, on the topic of brain stimulation is plasticity. So this is something that's less of a network phenomenon, more of a neural population level phenomenon, which is when I apply stimulation to a brain region in the context of a therapy for depression, or this is also used increasingly as a therapy for stroke recovery, what is going on at the stimulated regions in terms of induction of plasticity? Because we know that there are, and this is now repetitive stimulation, so repeated, basically oscillatory stimulation applied for around 30 minutes for maybe 5 days a week for a month is a common clinical kind of regime. But just for one of those bouts, right? So if you have a repetitive oscillatory stimulation applied to a part of the cortex for 30 minutes, then what is going on there at the level of the neural populations that result in long-term changes? Because that's what ultimately the therapeutic value is going to be coming from. It's going to be coming from long-term changes that are induced through some physiological plasticity mechanisms. Now, there are models and theories around specifically the type of plasticity that's induced when you have electromagnetic stimulation of this sort. Some of them study and kind of propose, understand this in terms of the equivalent of spike timing for rate models. Some of them invoke calcium transients, which is something that's relatively well understood at a cellular level. But what's novel is trying to model and understand this at the neural population level. But this broad area is another another kind of half of the picture in terms of the application of large-scale modeling in the context of brain stimulation. So the long-term effects that are resulting from plasticity induction. And again, this is an area that we're working with in my group, and we hope to have some interesting things to tell you about this in the future. It's early days yet. Great, so that's it for this lecture. I'll just want to thank everyone for listening, and I'll turn quickly to the questions. Okay, so we have 3 questions to go through here. Number 1 from Jamie. I think I read the entire connectome C. elegans has been mapped. Can you talk about the scale of this effort and why other animals have not yet to be mapped? Yes, absolutely. So C. elegans is the poster child of connectomics because it's been mapped, all 302 neurons. Why has this not been done in other animals? Well, because it's hard is the answer, but the extent to which it's been done in other animals. So the 2 kind of parts of that question are the resolution and the comprehensiveness. So I was talking about a lot of work in brain imaging that is, it's mapping the human connectome, but it's mapping it at a certain relatively coarse spatial scale, right? So we have coarse spatial scale maps of connectivity in terms of it's sometimes called projections rather than synaptic connections. So it's like an area connecting to an area rather than a neuron connecting to a neuron. So area to area projections and area to area connectomes, we have these for multiple species. We have these, again, at a certain level of coarse spatial resolution for, certainly for humans, I think all non-human primates of interest, and mice and rats and probably ferrets and others. And because anything you can stick in an MRI scanner, and if it's not too small, or anything that you can do a certain amount of chemical injection work, then those species have been mapped in terms of coarse scale connections. Now, in terms of the finer level of resolution, so yeah, C. elegans at the synaptic level is the only organism that's been mapped at that scale. Now, in the middle is the mouse, and that's the next one that kind of needs to be understood in this picture, is that thanks to the work of the Allen Institute, some other groups, but really driven by the Allen Institute, we have very high quality maps of the connectivity structure of the mouse. It's not all at the level of synapses. Some of it is at the level of synapses, and other people in the group can say a little bit more about it than me. But it's at a finer level of resolution than what we have from the level of projections from, say, neuroimaging. So at this point in time, the highest quality connectome that you can get in a species that is kind of interesting is the mouse, and it's from the Allen Institute. And definitely we can talk more about that, and Srijoy is the expert on these things. Srijoy's killing it with references. Yeah, thanks, Srijoy. It's a very long way. Yeah, good. Okay, next question. I always wondered whether it is, and this is Daniel. Daniel's been asking lots of great questions. I always wondered whether it's potentially problematic to use DTI to constrain connectivity analyses, because I believe DTI is especially bad in tracking long-range and sparse connections. But connectivity studies show that the sparse long-range modulatory connections are disproportionately important in terms of information processing, for example, small word properties, etc. What's your take on this? Right. This is a really great question. And so it kind of, right, it really depends what you think is the importance of sparse long-range connections. So it is true that, and it's diffusion, not DTI, diffusion-weighted MRI tractography doesn't do a great job with long-range connections, and particularly long-range connections that do things like traverse areas that have crossing fibers and traverse the corpus callosum, for example. So there's a lot of false negatives that come from diffusion MRI tractography reconstructions, but there's also, particularly if you use probabilistic methods, a lot of false positives. So it's a bit of a mess. So, you know, there's definitely a lot of caveats that should come from individual connection level descriptions, or anything you can, you know, look at the level of individual connections. But the broad statistical structure and the broad statistical spatial statistical structure, I think it's reasonable to say people will be fairly comfortable with being reasonably good at a kind of statistical level. One of the things that is not so good, and this is a real challenge with tractography, is, and we know this from particularly Henry Kennedy's work doing chemical tracing in the macaque, that the connection strengths vary over multiple orders of magnitude, like 5 orders of magnitude. So there are, and they also have this distance relationship. So really for connections that are separated by a long distance, they can be extremely weak in terms of like the ground truth. And this is something that diffusion tractography reconstructions really can't capture. They can't give you 5 orders of magnitude in connection weights. They can give you maybe 2 to 3, and a lot of that is introduced by measurement processes. So that's significant for, well, that's kind of, you know, one of the limitations of the type of thing that you were describing, is like sensitivity to extremely weak connections as a measurement technique. Now, in terms of what their influence is for brain structure, so all right, so your question about small world organization, it's an interesting one, and the thing is, it really depends what you're trying to describe. So if you're looking for the average behavior of the system, then you don't really care about the weak connections or the kind of small tail, right? So from that point of view, I think the intuition you get from small worlds, from the role of long range connections in small world networks, is a little bit misleading, because it's not clear to me, at least, that the brain functions in that kind of way. I mean, firstly, you know, the quantity that is described with, you know, in the Strogat's work, and just in general, when people talk about small world networks, they're talking about things like path length, right? How easy it is to wire information through the system, right? So for sure, like if you have a network and then you introduce a bunch of sparse network, you introduce a bunch of, you know, additional long range connections, then the path length is going to be decreased, right? But the type of things that I look at, they're really like average activity level type things. And it's really not clear that, you know, the average activity level is going to be kind of influenced in that type of subtle way that the graph theoretic path length is, right? So there's not, so you take like EEG signal or an fMRI signal, like it's not clear that having a single long range connection that dramatically decreases the path length of a graph would have any impact on the activity level of a neural population that's generating EEG signal. Like what that really cares about is the numbers, the average number of inputs, or the sum number of inputs, the sum number of outputs. So yeah, like it's a very interesting question, very on point question. It really depends where you want to go with it and what you're thinking about is important. And I have a lot more to say about this. I've thought about it a lot actually, but I'll leave it there for now. Okay, I know that vagal, okay. Jamie says, I know that vagal nerve stimulation can be used to treat treatment-resistant depression. Can this be modeled with these methods, or is it not really feasible because of the vagal connection to the enteric nervous system and the periphery? I mean, yes, it could be done. I'm very aware of the vagal nerve stimulation work. It's a really growth area in brain stimulation. There's a lot of interest in this. So, I mean, bottom line is it will certainly be something that people are studying one way or another. And in terms of how it fits into the models, so that's a bit more difficult because that's not part of the wiring diagram. So you'd need more information about like what are the projections of the vagal nerve, and how does that influence the activity at the level of imaging measurements, etc. And I don't know much about that, so maybe you can fill us in on it. But in general, right, what you're talking about is you're talking about introducing an input, and it's the same thing as kind of modeling sensory processing. It's like a simple way of describe like modeling a visual evoked response is you just inject a stimulus into the visual cortex, right? And it's really just a question of where is the input coming in. So the question in terms of, in the first instance, like modeling vagal nerve stimulation, although I'm not necessarily clear that this would be biologically what it's in. I actually don't know what the physiology of vagal nerve stimulation effects is, but the question would be where is the stimulus coming into the network, and then that's the starting point for addressing that question. Okay, thanks. Great. Well, that's the questions done. I apologize for running a little over time, but thanks everyone for the excellent questions and the attention, and I'll call an end to this session. Actually, before I call an end to this session, I just remember I had a request for an announcement. So I'm just going to do that. Yeah, so this, regarding the afternoon session, so why don't I flip back to the schedule as well, just to remind everyone what the plan is. So we have a lunch break up till 1 o'clock. And then we will resume with first tutorial from each side, then a break, then second tutorial from myself. We have some messages from the tech team, essentially, which is to update your environments. So there have been some updates, basically some bug fixes and whatnot to the Docker image and the KCNI school lessons tutorial documents. So if you have the environment set up and you already have this, Docker pull for updates and Git pull the repo for some updates in the code, and that should get you up to date for the session that will be the 2 sessions that will be going on this afternoon. If people have any questions or any of that's not clear, then let's please communicate with us immediately here and in the Gather Town and in the Riot Room, and we'll help you out. So I'll call the session to a close. Thanks a lot, guys. See you in the Gather Town.